It is my great pleasure and privilege to introduce Dr. Eric Green. Dr. Eric Green is the director of the National Human Genome Research Institute at the U.S. National Institute of Health, where he has been for nearly 30 years. During this time, he served as the Institute's scientific director for 7 years, chief of the NHGRI Genome Technology branch for 13 years, and founding director of the NIH Intramural Sequencing Center for 12 years. Dr. Green earned his MD and PhD degrees in 1987 from Washington State University in St. Louis, the same year the term genomics was coined. For over 2 decades, Dr. Green directed an independent research program that included integral start-to-finish roles in the Human Genome Project and groundbreaking work on mapping, sequencing, and characterizing mammalian genomes. Please join me in welcoming Dr. Eric Green.
Well, thank you very much. I am so excited to be here to kick off this symposium and to kick off this Science Week here in Toronto. What I thought I would do in my talk is really to set a context, in particular for this session, but also to really contextualize the significance of these awardees. It's going to be a little bit of a history lesson, very quick, and for those who aren't familiar with it, and for others who are familiar with the history, it's going to be a walk down memory lane, mostly emphasizing how did we get here and where are we going? I would contend that there were 2 scientific disciplines that were launched last century that really are changing medicine in this century. The word genetics was coined and first put into the literature in 1907. And then at about the halfway point of the century was the discovery of the double helical structure of DNA. And then, of course, very relevant to this symposium was, of course, the coining of the word genomics, as you heard in 1987, which was all part of an active discussion that took place throughout the late 1980s about the idea of comprehensively mapping and sequencing genomes, leading to the idea of what eventually would become the Human Genome Project. All of that reached an intense crescendo in 1989 because in 1989, 2 seismic things happened that changed the world forever. First of all, Taylor Swift was born, so we must keep that in mind. But second of all, the Human Genome Project was about to be born. And I got involved in the project literally about a year before in the late stages of planning, but then was incredibly fortunate to be a participant of this historic project that all of you now are familiar with that began in 1990 and continued on for 13 years, and I participated in it from beginning to end. And a lot has been written about this, and a lot is about to celebrate so many aspects of it. I do find myself, when I'm talking to younger people in particular, they tend to think we knew exactly what we were doing during the Human Genome Project, and that just couldn't be further from the truth. It felt the entire time like we were flying an airplane while we were building it, essentially. And all we were doing is just mapping away, sequencing away, and trust me, many times during the project, it was a very nauseating feeling of uncertainty if this was going to be successful. But at the end of the day, it was successful. And it was a remarkable achievement in human history and something that certainly should be celebrated. This organization certainly has celebrated it appropriately. I noted that awards were given shortly after the creation of the first methods for DNA sequencing, including the method by Sanger that got us through the Human Genome Project and generated by Sanger sequencing the first sequence of a human genome. But of course, in 2002, 1 year before the Human Genome Project ended, there was this remarkable group of people that were honored here in Canada for their contributions to figuring out how to map and sequence genomes and to get going on the Human Genome Project. But you know, it's 21 years ago that the Human Genome Project ended. It ended following a period of great, careful, critical, strategic planning that guided the project through it. And then there has been, ever since, in the last 21 years, what I would regard as a massive pivot. The Genome Project's completion was just really the finish line of one project, but a starting line of a new era. And it really was just the start when we got to that finish line. And what has been the pivot over the last 21 years has really gone in different directions depending on which different fields are using the tools and the technologies and the resources of genomics. I work at the U.S. National Institutes of Health. I have been the director for 15 years of the National Human Genome Research Institute. This was the institute created by the U.S. government to lead the U.S.'s effort in the Human Genome Project. And the reason it was embedded at the National Institutes of Health was we always recognized that what our institute would do would be to pivot from the Human Genome Project to the application of genomics to improve human health. This was not an incredibly surprising idea. In fact, when the Human Genome Project ended, you could have gone to the popular press or the scientific press and seen the juxtaposition of word genomics with the word medicine, because it was very clear that one application of genomics going forward was going to be to improve the practice of medicine. And so the notion of genomic medicine became the beacon. It became the thing that our institute knew we were going to be needing to lead, at least the NIH, the ascertainment of tools and technologies, applying them in medicine to improve patient care. There was just a slight problem. We could spell genomic medicine. We had no idea what it was going to be like. It was a very blurry concept. We needed to bring it into focus. That became a new journey that started 21 years ago. The starting line this time was the Human Genome Project. The finish line was going to be the realization of genomic medicine. And like the Genome Project, we weren't going to know every step along the way. But like the Genome Project, you do the first step and you check it off your list, and then the second step, and then the third, and you just keep working your way forward with your colleagues, and eventually you'll reach your destination. In order to do that, NHGRI, my institute, has led a series of strategic planning process and has developed a roadmap through 3 successive plans to try to get us through that journey and eventually realize the realization of genomic medicine. And so we simply looked at this journey and said, let's just deal with the first step. The day the Human Genome Project ended, we knew what that first step needed to be, and it was going to be a hugely important step. And in fact, we telegraphed it in the strategic vision that we published in 2003, the day the Human Genome Project ended, because we laid out what needed to be done in genomics for the first set of years beyond the Human Genome Project. And we said lots of things, and almost every one of them have come true. But the one relevant to this symposium was we talked about technological leaps that seem so far off as to be almost fictional, but which, if they could be achieved, would revolutionize biomedical research and clinical practice. And we gave, as an example, the ability to sequence DNA at costs that were lower by 4 to 5 orders of magnitude than the current cost, allowing the human genome to be sequenced for $1,000 or less. I was a co-author of this paper. I take a little bit of credit for that last sentence because I was trained as a pathologist. And at that time, I thought if we could get the cost of sequencing a human genome down to $1,000, that would be a very reasonable clinical test. The only problem was that we had just sequenced the first human genome ever, and it was coming in at a price tag of about $1 billion. And we therefore had to come up with some ideas in order to reduce that cost by 6 orders of magnitude and deliver what became affectionately known in the field as the $1,000 genome. What was that going to take? Well, one thing we knew was that the Human Genome Project and its $1 billion genome were done in factories like this. Expensive machines, not sufficient miniaturization or automation to get it down to $1,000. We needed something all new science fiction-like that would be the $1,000 genome. The good news is that the rest is history, and lots have happened, and we're going to unpack it, and the awardees are going to tell us about this, and so on and so forth. We kept really careful track of this. It's a very well-known, iconic graphic that we've been doing at the Institute for the last 21 years, very proud of our contributions to technology development in this arena, but it's been met worldwide by many creative scientists in both the private sector and in academic centers and government areas and so on and so forth, and we've gotten there. And that's been written about and celebrated, and it has truly, truly been an historically important technological innovation that have reduced the cost of sequencing a human genome more than 1 million-fold, allowing us to take that technology, those technologies, go out and sequence not one human genome, now millions of genomes, and we now know about hundreds of millions of places in the human genome, as one example, where we know how people vary. We have catalogs of those variants, and remarkable things have happened. This has been chronicled in the scientific and the popular press and celebrated widely. They didn't always get it right. And I just happened to dig out this slide, which I hadn't used for years, but the popular press occasionally didn't get it quite right with headlines like this, that soon it would cost less to sequence a genome than flush a toilet. I don't know what they were thinking, but okay, it was Business Insider. They didn't really quite get the metaphor, the one I would have used in any case. Besides just sequencing genomes, we had to interpret the human genome sequence. There we had it laid out in front of us 21 years ago, the 3 billion letters of the human genome. The Genome Project was never about interpreting all those letters. It was about peering over those letters, analyzing those letters, figuring out all the functional sequences. I spent much time reading the book of the human genome sequence. In reality, that's not the way you really look at the human genome. The way all of us look at the human genome nowadays is a richer and richer and richer view of the human genome through the internet that are based on unbelievable amounts of data that have been generated across all of the bases of the human genome, essentially giving us a Google map of the human genome. We've taken those technologies of sequencing DNA with an ever-growing understanding of how human variants influence human disease and human health, coupled with a focus on the functional landscape of the human genome, and we've been able to apply all of this to unravel the genomic basis of human genetic diseases. In particular, we've made striking progress in the case of rare genetic diseases. Do you know that when the Genome Project began in 1990, there were only 61 rare human genetic diseases that we knew what the gene was, and today we are nearly at 6,000 that we know exactly what is mutated in a patient's genome. That is remarkable progress. Now, most of the diseases that we need to be thinking about, especially filling hospitals and clinics within miles or within yards of where I stand, of course, are more common diseases. These are genetically complicated. They have many variants that are influencing what is often a greater contribution of the environment. But we're making progress, and there's lots of optimism that the next decade will bring us lots of insights about more common diseases. And so we can look out, and we could watch this journey unfold. And we've seen the cost of sequencing a human genome be reduced by more than 1 million-fold. And with that, I think we can cross this off our list. We have taken those technologies, and we have now sequenced more than 1 million human genomes, lots of information about genomic variation. We've peered over that sequence, and we've profoundly advanced our understanding how the human genome works. Not a full-size check mark yet. We have a long way to go, but what progress in 21 years. And we've significantly advanced our understanding of at least rare genetic diseases with lots of optimism. We'll learn more about common diseases. But what about changing medicine? That's the ultimate destination that at least my focus at my institute is. And here I can report that we do have some examples. It's a small check mark. It's the earliest examples. It is the tip of the iceberg. But we are implementing genomic medicine. Many of you are probably familiar with some of these areas. For example, in cancer genomics, it's completely changed cancer research. It is completely, slowly but surely, changing cancer care. There are many, many applications of genomics in oncology, and I think these will probably be the most impactful. We are seeing routine diagnostics nowadays for rare genetic diseases, either those that are just patients presenting for the first time with a condition, or patients who are acutely ill, like newborn babies, where we are now sequencing their genomes when the neonatologist doesn't know what is wrong with that patient in less than 24 hours, or patients who have been on multi-decade diagnostic odysseys, have undiagnosed conditions, and nowadays we sequence their genome, and in a remarkable fraction of the time we figure out what is wrong with them. The #1 genomic test worldwide is non-invasive prenatal genetic testing, where cell-free DNA of an unborn child is detected in maternal blood. This is done, estimated to be 7 to 8 million pregnant individuals will get that test done this year. We're seeing more and more information about pharmacogenomic testing, picking better medications of a choice of a list of medications for each patient based on their unique genomic signatures. And increasingly, we're seeing genomic prevention, whereby we can, in advance, test patients looking for known variants and have prevention rather than waiting until they develop a disease. So the bottom line is we have a lot to celebrate. We have seen remarkable advances in getting us closer and closer to the more large-scale mainstream implementation of genomic medicine, and we should celebrate. However, I also have to be realistic, and we are realistic in genomics. There are challenges ahead, and I don't want my exuberant optimism to let you believe that I think all this is simple or that any of this doesn't require a lot of careful consideration of many issues that we need to be pondering about. I sometimes talk about that genomics has now gotten to the adult table. I can tell you that during the Human Genome Project, and for many years, actually, for a short period of years thereafter, it was like being at the kids' table. It was so much fun. It was just a bunch of nerdy scientists like me and some of my colleagues just figuring out how to map and sequence the genome. We didn't have to think about the big problems. But, that sort of changed because, nowadays it's no, Eric, you can't sit at the kids' table. I don't care that they're talking about iPhones, Instagram, and Taylor Swift. You have to sit here and talk about mortgages, life insurance, taxes, politics, and genomic medicine in healthcare. Because once you touch healthcare, you touch society, and no matter what country I give a talk like this in, I know that healthcare is complicated and fraught with societal challenges. whether it relates to how this healthcare gets delivered, how it gets paid for, all the regulatory oversight that's so important to this, dealing with issues around equity and not exacerbating disparities because of the latest advances that we can bring in genomics that could potentially benefit all, but too often only benefit a few. Thinking about genomic privacy in line with a lot of other issues around health information about individuals and their privacy, and not to mention challenges around genomic literacy. This stuff's happening so fast. My medical school classmates, they never heard the word genomics once in medical school because the word didn't exist, and yet they're out there practicing and they have patients coming to them, some cases with already sequenced genomes, asking for information that might be relevant to their health. We have a lot of literacy issues to do to improve for both health professionals and for patients alike. Now, when we publish a strategic vision like we did in 2020, with our latest strategic vision, we think about these things. We talk about these things. We write about these things, both the exciting opportunities, but also the big challenges, including societal challenges, best exemplified by our Institute's Ethical, Legal, and Social Implications Research Program, our ELSI research program, which has been ongoing since day one of the Human Genome Project. So I encourage you to read the strategic vision if you want to hear what we think is going to be the next stages of this journey to the realization of genomic medicine for the rest of this decade. But I just thought I would, near the end of my talk, just tell you about something we put into this strategic vision that we never put in a previous one. We actually decided, and it may have been sort of because the tail end of the pandemic is when we wrote this and got it published, we thought it'd be fun to talk about the future, talk about what the next 10 years might bring. And so we put that into our paper. It's right there in the last text box of our strategic vision. There it is. We put out 10 bold predictions, and then Nature said, well, you have to refer to it. And so we referred to it in the text, and we said that now more than ever, solutions to these problems seem to be within striking distance. And towards that end, and with the characteristic spirit of genomics audacity, we offer 10 bold predictions of what might be realized in human genomics by 2030. Box 5. Everybody was so excited about these bold predictions. The press, colleagues, scientists kept talking about it, and yet I felt we didn't talk about it enough in our actual text of the paper. So I asked my communications group to put together a video that brings these 10 bold predictions to life because it tells us about the future.
Genomics, which is the study of all DNA in an organism called the genome, is changing the way we practice medicine, how we manage and treat some previously incurable diseases, and our relationship with society at large. Since the 1950s, double helix and DNA have become household words, standing in for the historic discovery of the 4 chemicals that give us life, commonly abbreviated as A, C, G, and T. Genomics is at an age of innovation and invention like no other, all thanks to how strings of those 4 letters encode the information for creating and operating something as complicated as a human body. Springing forward from the success of the Human Genome Project, a massive reduction in the cost of sequencing a human genome, and the dawn of the CRISPR gene editing era, genomics is leading into an even more audacious future. A future that will define our collective lives for decades to come. But what could this future look like? Here is a summary of 10 bold and fantastical predictions made by experts at the National Human Genome Research Institute and the genomics community for the next decade of human genomics. While most are unlikely to be fully attained by 2030, achieving even one or more of these by that time would have lasting impact for science, medicine, and society. Prediction 1. Sequencing and analyzing a complete human genome will become commonplace for any research laboratory. Prediction 2. The role of every gene in the human genome will be known. Prediction 3. Environmental influences on our genome will be routinely used for making predictions about health and disease. Prediction 4. Genomics will no longer use social constructs such as race in human research studies. Prediction 5. Student projects involving the study of millions of people's genome sequences will be regularly featured at school science fairs. Prediction 6. Genomic testing will become commonplace in medicine, just like a standard blood test is used now. Prediction 7. It will be readily known if a given letter difference in a person's DNA is clinically important. Prediction 8. a person's complete genome sequence will be available on their smartphone in a user-friendly form. Prediction 9. Advances from human genomics will benefit all of society and not just a few. Prediction 10. Genomic discoveries and technologies will help cure more genetic diseases than ever before. How do we get there? The National Human Genome Research Institute is committed to leading research to make such predictions a reality. Learn more about the Institute's 2020 strategic vision for the future of human genomics by visiting genome.gov/2020SV. Dive deeply into a field that continues to transform medicine and will hopefully continue to bring curiosity, purpose, and compassion for people everywhere.
So I hope you enjoyed that fanciful and a bit metaphorical set of predictions, but we think in many ways it charts a course of the kinds of things we want to be achieving for the rest of this decade. So I have quickly told you about the remarkable progress in the last roughly 2 decades since the end of the Human Genome Project. This pivot that we made when the Genome Project ended has, if nothing else, has begun to bring into focus what genomic medicine is going to be about. I certainly think the examples to date have been inspiring, but I would be equally confident in predicting that some of the things we will see in the next 10 years will be equally, if not more, inspiring as we see more and more applications of genomics in medicine. In closing, I would also say that I stand here getting to represent a remarkable community of scientists, first who figured out how to map and sequence the Human Genome Project, although it was a very small community of scientists, all new to the field, all coming together with a very laser-focused set of goals. But today, and what we've seen over the last 21 years, is this incredible growth as we've seen genomic medicine be realized with individuals from multiple disciplines standing on each other's shoulders year after year. This group is remarkably talented, it's increasingly diverse, it's certainly international, and it's incredibly interdisciplinary, and I am honored to be a part of this community and to watch them make accomplishments that I once upon a time never thought was possible. So thank you for your attention, and if there's time, I'm happy to take any questions. Thank you very much.
Thank you very much, Dr. Eric Green. That was really good to know more about the background of human genomics, but also your thoughts on the future of it. We do have a question. The question is, you compare the human genome to building a plane. What was the biggest obstacle you faced early in the project?
Remarkable strategic uncertainty. And I think very relevant to the awards that are being given this year there was absolutely the belief, and it's written about in some of the documents leading up to the Human Genome Project, was that we would dare not start sequencing the human genome until there was a revolutionary new sequencing method. The idea was you could take Sanger sequencing out for its test drive on the model organisms with smaller genomes like flies and worms and bacteria, but we would wait until there was a revolutionary new method. And it was incremental, incredible improvements in Sanger sequencing that were cobbled together that got us to the point they decided to hit the accelerator by about 1996, 97 to start sequencing human. But it was a lot of uncertainty at that as when that pivot was going to go from mapping to sequencing, at least in the case of the human genome.
Okay, perfect. Thank you. And for those in the audience, please use the Slido as much as you can. And in the meantime, I do have one question. You mentioned 10 bold predictions. Is there one that you're most excited about?
I would probably, so that's like asking me for my favorite child, because that would get me in trouble, because every one of those 10 represent areas that we're working at the Institute. Yeah. I will tell you that as somebody who's trained in clinical pathology, I particularly like the one where it talks about that we will just regard a genome sequence as routine as a CBC, a complete blood count. Because to me, it's, growing up in medicine, as medical school, CBC was just a routine thing. Everybody got a CBC. I would love to see everybody just have a genome sequence. So that would probably be my favorite. Although the one that gets the chuckles the most, having shown this video, is the idea that we're going to access our genome on our smartphone. People seem to chuckle when they see that.
And while we're waiting for a bit more question, I do have one more. You mentioned about with the bold prediction about the environmental effects, and I think that's a very big one because we think about genomics and environment as a separate entity, but trying to use genetics to explain environmental effect. Can you expand a little more on that, please?
Well, I often talk about, and partially because of these guys here, is that the genomics has just been so lucky because we got lucky with our technical innovation. We had these next-gen sequencing methods come out that allowed us to ride this incredible wave of data generation and in some ways unfairly get way ahead of the other determinants of health because we're studying the genomic side of the equation. But, you know, I firmly believe that there are already or coming and will continue to be innovation in the space for generating huge amounts of data about environmental exposures. And that data will be matched by our genomic data, and the next big advances will likely bring those data sets together and allow us to start to really do the kinds of studies we should be doing for the most common health conditions that are affecting humankind. So I just brag about the fact that I've ridden a wave of technology innovation, but by no means do I not think the other part of it is important. I'm just waiting for their technical innovation, which I'm sure is coming.
Perfect. So more data is coming.
Yes.
And we do have questions from Slido as well. Do you predict a similar pathway of development to the field of epigenetics?
Yes, fair enough. No, absolutely. And, you know, once again, how lucky and fortunate is it that we've seen technical innovation build on the core technologies of next-gen sequencing to be able to read out epigenomic marks. I'm sure we'll hear about this from some of the other speakers. But no, absolutely. And in a longer version of this talk, I unpack that a little bit more, talking about, you know, these other omics, and certainly epigenomics is among those that clearly there's going to be a lot of exciting development and a lot to unpack because there's probably a lot we don't know.
Okay, perfect. I think that's it. Time for your
Okay,
questions.
thank you very much.
Thank you very much.